really special. Okay, it's very special for me to, even from far and a different continent, to be with you in Africa and in Cape Town that I never been. I've been in North Africa, but never in the South. So I hope one day I will have a chance to visit your beautiful country. And today I will talk to you how my lab is actually exploring the cell division of the parasites using what we call not a different type of microscope, but a very specific expansion microscopy that we use and call ultrastructure expansion microscopy. And so before, I just want to let you know what my lab, who were able to actually do the science. Some of it was done by me, but everything is actually a collaborative effort. And I really wished that everyone in the world understand that we can't defeat malaria if we're not working all together. So some of the data you're going to see was actually in collaboration with my postdoc advisor, Dr. Voin, that he works at Harvard Medical School and Boston Children's Hospital. He's a pediatrician as well, so he understands very well the burden of malaria for every family that is suffering from. We also have my star postdoc, Ben Liefner, that's going to open his lab in Australia and continue to the fight against malaria, my graduate student James, and amazing collaborators at the NIH with Joel Vega Rodriguez and Tiago, and also a very international collaboration. We have a team in Portugal that have been fighting malaria for decades in the Maria Motas lab. So in my lab, we're actually a very basic biologist, which I think we are also part of the fight. We don't do drug development, but if we don't understand how the parasite is actually replicating, it limits the drug target we could think of, and our imagination can go further to develop new treatments. So we do believe that more we know about how a eukaryotic cells is built or divide, we can actually understand better how we can defeat this parasite. So we're really interested to understand key molecules that build this very amazing, as a cell biologist, eukaryotic cells, and also what are the mechanisms at the molecular level that help to dispatch all these organelles that the parasites use to invade and replicate in the host, in both the human and the mosquito host. But then when you think about the tree of evolution and what are the models we use to understand the eukaryotic cells and our eukaryotic world, it's mostly coming from one very, very specific phylum. And I do believe that it's not because it's the most magical, interesting one. It's, I think, because we belong to that phylum. And so I really want to try to emphasize that we have to look at the other side of the evolution tree. And one of my favorite phylum is the alveolata, where actually Plasmodium falciparum belongs, but you don't have only Plasmodium parasites in that phyla. You have other parasites that are causing human and livestock diseases. For instance, Toxoplasma is one very well known for human diseases, but Theileria and Sarcocystis in the livestock are really important. And all those amazing as a cell biologist, not as a human being, I do not appreciate them, are amazing in the way of their way they divide their nuclei and the way they actually do cell division. And they do it in a very divergent way. You can see the examples and the names that we give to those mechanisms. And I'm not going to go into details of how they do it, but because they are so divergent, we can exploit that divergency to identify new drug targets. And it's really, this is actually one of the first picture I want to show you. It's how the malaria parasite looks like in the human hepatocytes. And really it starts in the early division by being a bag of nuclei here in blue. That's what you see, the chromatin, growing into a vacuole that is here stained in yellow. And somehow they gain polarity. And then in 12 days, you pass from one parasite that infects an hepatocyte to 100,000. The question is really what those factor mechanisms that result into this sophisticated cellularization when we come from, on the left side, of very disorganized cells to the right side, where we actually know that dozens of sub-organelles has been created and organized to make this parasite efficient to invade. Now, when you think about this phylum that we call epicomplexan, because they have a complex apical end, they all field of organelles that are very important for actually invasion and egress and the development of that vacuole that protect them from the host. But the issue is when you want to study that malaria parasite that you have here with a scale bar, and that's where we're going to talk about imaging. This scale bar is telling you that most of those organelles are sadly in lower than in the best resolution you can get from an optical microscopy, right? Which the best channel will give you a resolution of 200 nano. Resolution means that I can only distinguish things that are distant from 200 nano, which is actually a quarter of that parasite. So not really helpful. And that's where the idea of expansion microscopy in the field of cell biology is so amazing. The idea is really, instead of looking closer, and I'm sorry for Zeiss company, but really to not buy your amazing lattice light sheet microscope that costs still a lot of dollars in the US, for instance. The idea is really, why don't we make it bigger? And so this image is actually a late stage Schizon's parasites that looks like in your red blood cells about to egress. And what you see in blue, again, it's the chromatin of the parasite, so it's a multinucleotide. And then in magenta, it's actually microtubules that you have in the cytoskeleton. Now, that's what I was able to make using actually the SIM microscope from Zeiss, amazing super resolution microscope. But it's still, if I'm telling you that those subperigram microtubules are between 2 to 3, you're not going to believe me, right? But now if I take the same picture with this time the LSM 800, so with the Eriscan, not even needed the SIM, now you can believe me and see that those microtubules are indeed up to 2 or 3. You even have a teeny one here that it's actually really interesting. And you can see from where they start to where they end. And it's because the resolution now are increased. So before in that channel, I had 298 nano resolution, but thanks expansion microscopy that I expand my samples up to 4.5, which mean in volume 100, actually in volume, my resolution is 960 nano without using a super fancy storm or any type of super resolution microscopy. Now, the life cycle of the parasites that is between the host, the mosquito, and the human. Before, cell biologists will look at those beautiful parasites this way. Now, with the lens of expansion microscopy, that's how we see the parasites through the life cycle. And you can already see so many details. Here is actually an oocyst. So the cells that grow into the wall of the on top of the midgut wall in the mosquito, and in 12 days, one parasite will develop into hundreds of thousands sporozoites that are the ones that the mosquito actually inject in your body. Now, I want to give a few minutes to really explain what is expansion microscopy for the people in the room that are not familiar. It's really a sample prep. So it's not a new microscope, it's a new sample preparation for microscopy. So you basically start as everything you do I'm sorry, I have an issue with my mouse. You really start like you always do for immunofluorescence assay. You start to fix your samples. So we usually do PFA or glutaraldehyde fixation, but actually, if you're interested to look at membranes of your samples, you should go for cryofixation. Then what we do, and that's where the magic actually, we still don't understand at the chemical level. So don't ask me, we still don't know. That might be the next Nobel Prize. Is we place a solution made of formaldehyde and acrylamide into our samples, and we call it the anchoring solution. And that anchoring solution will really allow all biological molecules such as DNA, proteins, and lipids to anchor into a gel. And that's the beauty of that technique, a gel that is a swallowable gel that will expand into contact of water. Why? Because that gel is made of sodium acrylates. That may not call you anything in your mind, but it's actually what you found in diapers. So this molecule is the most expandable molecule. We know the properties, and in contact of water expand. And then what is exceptional with that technique is your biological sample embedded in that gel will be able to expand in water. Why? That during that expansion, all the isotropic volume of the cells is maintained. So you have no distortion of your samples. So we do a very specific denaturation that is soft, 95 degrees C, 90 minutes. Then we expand our gel, shrink it to do the staining, re-expand before going to the microscope. And really, I want to emphasize that we in the lab use your beautiful Eriscan 2 microscopy, the LSM 900. You cannot steal, sadly, the parasites is so small from the beginning, 1 micro. We still need the beauty of Eriscan 2 to get sub-resolution images of our expanded parasites. But if you were working on mammalian cells, you could just go for confocal microscopy. But I have to say, Eriscan 2 is actually really amazing for us. And I will also detail why Eriscan 2 rather than Eriscan 1. So that's how we see now our blood stage parasites. And one of the amazing actually developments of expansion microscopy is the use of dyes that before was used more as a biochemistry tool to pull down proteins. So one dye that we really use intensively in the field now is called NHS ester. And what NHS ester does, it binds all your amine groups. So it will bind all the proteins of your samples. So what you see here is not an electronic dense image obtained by electronic microscopy. It's actually a protein dense map of a late schizon, so a late stage blood stage parasites in the red blood cells. And what you all see here is actually the organelles that compose the parasites. And unlike mammalian cells, people working on mammalian field, I cannot go online and buy an antibody against every single organelle of the parasite so I could study it. One, because companies do not produce them. Two, because most of the time we actually still don't know what are the protein composition of those organelles. So as a cell biologist, I can tell you, I can recognize by shape those rock trees that are the invasive organelles of the parasites. Very important for the parasites to be able to bind to the red blood cells and invade. And then the little granules, we know that they are macronyms and exonyms and dense granule. We can even, thanks expansion microscopy, define which protein in which granules. And those protein MA1 and EBA are actually vaccine candidate for malaria, against malaria for decades. So now we are going to have a tool to understand better when they are secreted and what is the outcome of those proteins to maybe better design vaccine and drug development. Now we even understand better the function of those rupture. We know there are fields of enzymes that are essential for invasion, but in collaboration with a team in UGA in the US, we actually proved that one of those proteins, the RUN11, is actually essential for having 2 ruptures. And we were surprised to discover that 1 rupture is not enough for the parasites to be invasive, which is amazing. Now we don't need to completely kill the ruptures. We just have to actually reduce it by half, which makes you hope that sometimes treatment, if they are not fully efficient on the parasites, could be efficient in a patient. Then our lab is really trying to expand, sorry for the kind of inside joke, the use of that technique. And what you see here is actually a picture of our LSM 900 with a beautiful 40x water objective with a piece of gel. And inside what you see is actually the entire midgut of a mosquito that we are expanded. And each little ball that you see on your right side is actually an oocyst that will develop into 100,000 of sporozoites. And now I'm looking at you, before you were seeing in 5x, now I use my 40x objective water and shows you how the parasites is developing in each little oocyst. So now for all the biologists and drug development labs in the world that try to develop new drug that mosquito will be sensitive and affect the development of the parasites, you have a better tool to look at really the development and the effect on the development of the parasites. Because before, people will measure the diameter of the oocyst to infer the development of the parasites, but I'm sure you can by yourself realize that you have different stages of development, and it's quite the same size for some of those oocysts. So I think we need to revisit the data in the field, and now you have better tool to really look not only at the parasites, but at the effect of the drug on the host mosquito as well. And so this work was only possible with colleagues like I mentioned, Joel Vega and Tiago. We also did it for the salivary gland. And now you can actually see in here in pink, the parasites going through in 3 dimensions of the salivary gland. Which again, if we go back to the RUN11, we have learned now why as well this protein is essential at the mosquito level. So if you have a wild type parasite, what you're going to see on the left, it's 2 parasites. One in with the yellow arrow is showing you that it's already in the secretive cavity and can already infect in a human. The other one in the green is still in the epithelial cell. Now, if we look at the RUN11 mosquito, RUN11 infecting the mosquito salivary gland, you could before just so see that you have less parasite in the salivary gland. But what we know now is that the parasite is stuck between the cells. It really can never get into epithelium cells. It really stays between them and then will never reach the salivary gland. So really this RUN11 protein is kind of a very attractive protein now to even block transmission of the parasites. So not only we were able to understand better the biogenesis of those organelles, but we also identified now the mouth of the parasite, and that's what we call the cytostome. What do we care is because before the only way to see this mouth, and that's the way the parasites take the hemoglobin, was electronic microscopy. And then why we care is because sadly the resistance of artemisinin drugs, that is our first frontline drug for malaria, and we know we have resistance since 2010, and now it's actually spreading. We already not spreading. I don't want to be alarming, but we're already detecting resistant parasites for artemisinin in the African continent, which is very alarming to us. We against Kel-13 protein that is at the neck of that mouth. Now we have a tool that it's not electronic microscopy to really follow what makes that mouth changing when you are resistant. Not you, the parasites are resistant to artemisinin. And in collaboration with an amazing researcher that I'm sure you all know, Dr. Diane Worf at Harvard, we really work now on understanding better the hemoglobin uptake so we can try to infer better the how the parasites resist artemisinin. And we actually found a new mutation in parasites in Senegal. It's on the choronin protein, and we could not understand before we were using expansion what was the role of that choronin, and now we know that it's involved as well in uptake of hemoglobin. So now we know there are 2 pathways that are involved for hemoglobin uptake by that parasite. So if you're more interested to know with all the organelles we discovered through and we looked at through expansion, please go to our recent publication that were led by 2 amazing scientists, Dr. Liefner and Anna Carla. And I think I'm running out of time, so I'm going to pass that slide and then just thank you for your attention, and I can take any questions.